Today, we're going to be focusing all on your sense of sight. Let's get started. I've been on a grind with these 3D anatomical models, so why stop now? I present to you, in all of its glory, the beauty that is the human eye. So, for you to be able to see, the first thing that happens is light waves are going to pass through your cornea. So, how about we hop on some light waves and ride right on into the human eye? Let's get ready. So here we are inside the human eye. Let's start learning. We just traveled via light waves through the cornea. This is the clear front layer of the eye. The cornea is going to be shaped like a dome in order for it to bend light to help the eye focus. Some of this light is then directed towards the pupil, while the iris controls how much light the pupil will let in. The iris is going to be in charge of controlling the diameter and the size of the pupil. The iris is very easy to locate in the eye. It is simply going to be your eye color. So whenever anyone comes up to you and says you have nice blue eyes or whatever, they are really complimenting your iris. The pupil is a hole in the center of the iris that allows light rays to enter the eye and strike the retina. After passing through the pupil, light passes through the lens, which is the clear inner part of your eye that works with the cornea to help correctly focus light on the retina. And since I've mentioned it multiple times already, our retina is going to be a light-sensitive layer of tissue located in the back of our eye. Inside the retina, millions of sensory receptor cells, known as photoreceptors, are hard at work. These photoreceptors are going to take the light waves projected onto the retina and transduce them into a message that the brain can understand. We have two different types of photoreceptors that help us sense color. Rods are tall, thin photoreceptors that are responsible for detecting black, white, and gray, and are necessary for peripheral and light vision. Cones are going to be shorter, rounded photoreceptors that detect fine detail and are responsible for color vision. Located in the retina, we also have our bipolar and ganglion cells. Preliminary processing of visual information starts with the ganglion cell. Our brain contains about 1 million ganglion cells and over 130 million photoreceptors. As you might think, this would be a very hard job to take on single-handedly. Luckily for our ganglion cells, the bipolar cell is here to help. The bipolar cell collects information from our photoreceptors and funnels them into our ganglion cells. This neural message is then passed along the optic nerve and onto the thalamus. Fun little fact about the optic nerve: the area where the optic nerve leaves the eye has no photoreceptor cells present. This will actually create a blind spot in our field of vision. I've posted a link in the description box that will bring you to a website to find your blind spot. After the message has traveled through the optic nerve, it reaches the thalamus, which will then send it to the appropriate brain structure. In this case, since we're dealing with sight, the occipital lobe. So, that explains how light waves enter through the eye and are transduced into a neural message for our brain to understand. But how exactly do we explain how we sense and perceive sight the way we do? Before we answer those questions, let's head back to the classroom. Alright, welcome back. So there are going to be two major theories that attempt to explain our perception of color. The trichromatic theory of color, proposed by psych team duo Thomas Young and Hermann von Helmholtz, states that humans perceive color because there are 3 varieties of cones in the eye sensitive to different wavelengths. These wavelengths being blue, green, and red. Red light produces long wavelengths, green medium, and blue short. According to the theory, different light waves activate different cones. There will be some overlap with these cones, and you will find out why in just a second. When you look at a good old normal red apple, none of that Granny Smith nonsense, the red wavelengths strike the retina, activating the red sensitive cones, allowing us to perceive red. Now, you may be thinking to yourself, well, I can see more than just red, green, and blue. What's up with that? Well, I will tell you exactly what's up with that. When a color other than red, green, or blue strikes our retina, it will stimulate a combination of cones in order for our brain to perceive the correct color. For example, if you look at a lemon, the yellow wavelength strikes our retina. Then, red-sensitive and green-sensitive cones are both activated in order to produce a neural message of yellow. When trying to remember the trichromatic theory, just look at its name. The prefix tri means 3, just like a tricycle has 3 wheels. The trichromatic color theory states that 3 different color-sensitive cones in our retina allow for our perception of all the colors we see. In the late 1800s, arrival theory, known as the opponent process theory, was proposed by German physiologist Ewald Hering. Hering questioned why individuals who were colorblind to red and green were still able to see yellow. According to the trichromatic theory, we do need active red and green cones in order to perceive yellow. Hering attempts to explain his theory by using after images. An after image is going to be a visual experience that occurs after the original source of stimulation is no longer present. To better understand the idea of an after image, I'm going to run a little experiment. It's going to be a very simple experiment. All you have to do is stare at a dot on a screen for about 20 seconds. Those are your only instructions. Stare at the dot and let the magic happen. Here we go. trichromatic theory only explains how color vision happens in the receptors, but not how we perceive colors in the brain. The opponent process theory states that opposing pairs of color receptors enable color vision. So red-green, yellow-blue, and white-black. A century after Herring proposed his hypothesis, scientists were able to confirm it. In the retina and thalamus, we can see that some neurons are excited by red but inhibited by green, or maybe even excited by green and inhibited by red. So how does this explain our afterimage? When you were staring at the picture of the building originally, I had the colors inverted. You saw a bunch of different sort of shades of blue and purple. Well, this actually excited several neurons in the thalamus and retina. After continually being exposed to this stimulus, we eventually tire out our blue sensitive neurons. So when I switched over to just the white image, and since white includes all of the colors, all of our other color sensitive neurons will fire normally, while our overstimulated blue sensitive neurons will need time to recharge. As a result, we see our afterimage. Today, the understanding we have on how we process color goes something like this. The retina's red, green, and blue cones respond in varying degree to different stimuli. Then the message is processed by the nervous system's opponent process cells before starting their journey to the occipital lobe, proving both the trichromatic and opponent process theory useful in explaining our color vision. Our last topic of discussion is going to be sensory disorders involving vision. We will start off by talking about nearsightedness and farsightedness. Your acuity or sharpness of vision can be greatly affected by small distortions in the shape of your eye. In a normally functioning eye, the lens focuses the image of any object perfectly on our retina. Nearsightedness occurs when a physical distortion of the eye causes the light rays from distant objects to focus in front of the retina. With nearsightedness, you're able to see near objects a lot clearer than distant ones. Farsightedness is going to be the opposite of nearsightedness. Farsightedness is when light rays from near objects reach the retina before they have time to fully focus. A cool fact about our eyes and the brain is that generally we are able to accommodate and compensate for the unfocused image, making it so those with farsightedness rarely need glasses. However, it can lead to eye strain from the overuse of the eye muscles, as well as headaches. People generally do not discover that they are farsighted until around middle age, when the lens loses its ability to change shape rapidly. Color blindness occurs when people are unable to perceive color in a normal way. Color blindness is most commonly passed down genetically, but any damage to the retina or optic nerve could potentially lead to color blindness. It's going to be caused by a difference in how the photoreceptors respond to certain colors. Various pigments on the cones, known as photopigments, make it possible for us to see all visible colors. If there is a problem with these pigments, your eyes will experience issues with seeing colors the way they should. Cortical blindness occurs when partial or total loss of vision is a result of damage to the brain's occipital lobe. With cortical blindness, the structure of the eye is perfectly healthy and doing its part, but once the brain receives the message, it cannot interpret it. This type of blindness is also known as cortical visual impairment, or CVI, and is one of the leading causes of blindness in children. Age-related macular degeneration, or AMD, is another leading cause of vision loss, affecting an estimated 10 million people in the US. Macular degeneration occurs when a small, tiny portion of your retina, known as the macula, wears down over time, leading to a loss of vision. Well, that does it for our vision. If you enjoyed the video, feel free to like and subscribe to the channel. This will notify you whenever I put a new video up. Anyways, I will see you guys next time. Peace.